So, good afternoon, everyone. In today's class, we are going to discuss about the excretory system. So, the chapter name is excretory products and their elimination. So, we all know that in every organism, those who are living, they will be have the excretion in their body because you just imagine our body utilizing the oxygen and will do many metabolic activities. So by the metabolic activities, the waste products can come up. So how we will do the metabolic activities? That will be happen by the intake of the food. So if your food intake is too much, that food itself can be a waste product because body cannot utilize the excess food intake. So the first thing what we are going to see is how the waste products will be formed in our body. How the waste products formed in the body. So the one thing I already said that the one reason behind it, the metabolic activities. So the main reason behind the this excretions or the excretory products in our body it's because of the metabolic activities. By the metabolic activities, many things will be, many excretory items will be formed in our body. For example, you know that the sodium, potassium, urea, uric acid, ammonia, etc. Now, the second thing I already told you that how that will be formed up. It also can be formed up. This excretory products also can be formed up by, or this waste products also can be formed up by the excess intake of the food. Right? So in that way also it can be formed up. So we have seen what are the two reasons that will bring the waste products in our body. The one, it is metabolic activities. You know that every organs will do the metabolic activities. The tissues, you take the any kind of tissues, they will utilize the oxygen, and one of the very good example I can tell you, the carbon dioxide is the byproduct. So like that, if you take a piece of chicken, the whole piece of chicken we cannot utilize. Many proteins, many fat, many carbohydrate we can take from that, but again, we will get some waste products from that. So in that perspective, if you're looking, this waste elimination in our body is one of the very important functions. Because otherwise, what will happen? Whatever the waste we are getting through the metabolic activities and the excess intake of the food, it will be accumulate in the body and it become toxic, and that can become, or that can be lead into the death of the person. So that's why the excretion, this excretion in our human body and the other organisms body is really, really important. Now the point number two, what we are going to see is, listen, here what we are going to discuss. I already told you that there are different types of waste products. But in this chapter, the excretory products and this elimination chapter, mainly we are focusing on nitrogenous waste products. right? Mainly we are focusing this nitrogenous waste products, how they will be eliminated from the body. That is our main focus. Which are the main nitrogenous waste products? They are ammonia, urea, and uric acid. Because this is the major waste products that will be formed in our body. So that's why we are giving main focus into that particular waste products, the nitrogenous waste product. Because why we call them as nitrogenous waste product? Because all these products contain the nitrogen compound. So that's why we call them as the nitrogenous waste product. That is also clear. Now, while we're looking on to the next one, everybody please look into the next part. The next part, what we are going to discuss. If you're taking the different organisms, for example, some animals living in the water, some animals living in the terrestrial conditions, some animals like the small animals or the some other animals like the snakes or the apes. So that kind of animals, if you are taking, all these animals' excretory system is entirely different. It's not very similar. So that's why the excretory products also will be different. So the where they are living, what kind of excretory system they have it, based on that, the major nitrogenous waste product excretion also will be different. So let me make it very clear. First of all, if you consider our human body, please understand in our human body, please understand there will be the ammonia formation, there is a urea formation and the uric acid formation. There is no doubt in that. Everything will be formed up. But if you're looking in our body, you will realize one thing. Whatever the ammonia formed in our body, that will be converted to urea, and as a urea, that will be excreted from our body. But doesn't mean that ammonia is not forming in our body. Ammonia is formed up. But ammonia is very toxic to our body. That's why never it will be excreted as ammonia. The ammonia will be converted to urea and will be excreted out from the body. So now the next step, the next session, what we are going to discuss it is what are the different types of excretion based on their excretory product. So that's what the thing we are going to discuss. Everybody listen to that. And by the time I will detailedly discuss about the ammonia to urea conversion. First of all, we can talk about ammonotaric. So everybody listen here. What is the ammonotaric? The ammonotaric means what? The organisms, their major waste product will be ammonia. So organisms, their major waste product, it is ammonia, the major nitrogenous waste product. That kind of organisms only we call it as ammonotaric. So what we are discussing here, what are the different types of the excretory system or the excretory waste, types of excretory product. The first one we are discussing here, it is ammonotaric organisms. The organism, their major waste product will be ammonia. So here, what you have to understand, what are the character the organism should be have if he want or if she want to excrete the ammonia from his or her body. The major character he should be have it most of the time. That organism should be have the good availability of the water. That's a next first thing you have to remember. The water should be available. The availability of the water. Why the water is required for the ammonia excretion? It's very important. So listen everyone. The reason behind it is ammonia is readily diffused into the water, or they are readily soluble in the water. The number one thing why the availability of the water is very important for the ammonotaric organisms, because the reason number one, ammonia easily soluble in water. That's the number one reason. Okay. Then, if you want to excrete out the ammonia, we require more amount of the water. Requirement of more water. So these two criteria, you check it. Ammonia is easily soluble in the water and the requirement of more water if you want to excrete the ammonia. Where this kind of organisms will be there? Your answer will be very, very simple for that. The organisms living in the water. That's a very immediate answer you can give for that. Absolutely right also. Because the availability of the water is much more in the sea or in the other water regions. So those organisms fulfilling these two criteria, they can have this ammonotaric excretion type. So what will be the example for that? Some examples are some bony fishes, the aquatic amphibians, aquatic insects, etc. They have the good availability of the water, so they can have this ammonotaric excretory system. No problem with that. Now, the question you may have to ask here, what is the one question we will really think about it? How this will be easily possible? That question also we have to answer, because simply the availability of the water, how it will be possible? That's we have to see. So please look on to we can just take the case of fishes. You take the fishes. Right. Take some bony fishes, you can take it. The ammonia can be excreted out through way. One, through the body surface itself. The one more mechanism it is through the gills. So listen everyone, what we written here, some bony fishes, aquatic amphibians, they have this ammonotaric kind of excretion. So what are the organ structures will help for that? One is gills, another one is body surface. So in these two structures, through this gills and the body surface, by the diffusion mechanism they can excrete out. By the diffusion mechanism. Here, that is also very important. So no doubt in that. So we are talking about the types of excretory products or the excretory way. The first one we have discussed, it is ammonotaric. You understood what is ammonotaric? The organism major nitrogenous waste product will be ammonia, no doubt. So the requirement, the criteria we discussed, examples we discussed, the structure for this ammonia excretion also we discussed. That's very clear. Now we can, in the types of excretory product, we can move into the next part. That is the next type. It is ureotaric. So ureotaric, listen everyone, Ureotelic means from the word itself you can understand the excretory product will be urea. Right. So we can start with the examples. After that, I will go with the other details. The major examples for this, it is humans or mammals, then marine fishes, terrestrial amphibians, these all are coming under this ureotelic. Here you have to understand too many things. Why the humans? Humans case we have to discuss little more. Why the humans are in the ureotelic mechanism? You have to clearly understand that. How the urea formed in our body, that mechanism also you should be clear. So let me talk about it first. So how the urea will be formed in our body? It's very simple. Urea mainly we are getting from ammonia convert to urea, or the main step if you want to get the urea in our body, that is the reason behind it is ammonia only. Then the question will come, how the ammonia will get into our body? Ammonia will get into our body by protein metabolism. Whenever the protein broken down happen, immediately the ammonia will be formed up. From that only we are getting the ammonia will be converted to urea in the liver. So listen everyone, that point we can discuss a little more. So if you just imagine, all the time you have to remember one concept. In the case of fat and the carbohydrate, we can store the fat and the carbohydrate. But in the case of protein, we cannot store the proteins in that form. You can store the carbohydrate in that form, you can store the fat in that form, but never you can store the proteins in that form. So the metabolism of the protein will be comparatively higher in the case of human body. You have to please understand that concept very clearly. Now, let me talk about it. First, how the ammonia will be formed, because from that only urea we will get. How the ammonia formed up? The ammonia mainly formed by protein metabolism. Right. Now, you take any tissues, ammonia will be formed in the tissues, no doubt in that. Every tissue you take it, by the protein metabolism, ammonia will be formed up. So please understand, first of all, everybody please listen. You take any tissues, this tissue can be nervous tissue, liver tissues, cardiac tissues, any tissues you take, there ammonia will be formed up. So what will happen immediately? This ammonia will be trapped by glutamic acid. Then the glutamic acid, they will be converted to glutamine, right? And they will go to the liver. There it will convert to ammonia again. Then in the liver, this ammonia become the urea. This is a mechanism happen actually. So in the tissues, ammonia will be formed a lot, no doubt in that. It will be trapped by the glutamic acid, it becomes glutamine, then it will goes to the liver, again it will convert to the ammonia, then it will be converted to the urea by many mechanisms. So remember everyone, ammonia as it is very, very toxic. We cannot keep the ammonia for more than 5 minutes or 3 minutes or even maybe 10 minutes. We have to make it immediately, we have to metabolize. So that's why this trapping mechanism will happen. It will goes to the liver. The ammonia again will be converted to urea because this ammonia is very toxic and very sensitive to our neural system. So if you allow to retain the ammonia for some time, we may get the neural toxicity also because brain cells are extremely toxic to extremely sensitive and toxic to this ammonia. That point also please remember. Now I would like to tell one more thing. Actually, what you have to remember here, the one more point you have to remember. The one point you have to remember here it is. I told you that by the protein metabolism only, you will get the ammonia and the urea. This urea formation, that cycle, we call it as urea cycle. The urea forming cycle, what we call urea cycle. So this urea cycle, we will have the two names. The urea cycle also we can call it as Ornithine cycle. So why we call them as Ornithine cycle? Because ornithine is the first member in the urea formation. That's why we call it as the Ornithine cycle. You can also call it as this, based on the scientists who discovered it, you can also call it as this cycle, also we call it as Krebs-Helmich cycle. That is another name. So the urea cycle is otherwise known as Ornithine cycle. Otherwise, based on the discovery of the scientists, we can also call it as the Krebs-Helmich cycle because these two people only discovered that. I hope it's very clear. So, remember what the point we discussed. Ammonia is formed in our body, but as it is highly toxic, we cannot retain as ammonia, so we should immediately convert to urea. So that process will be happen inside the liver. And that cycle names I have written. This urea cycle we call it as Ornithine cycle, or as we call it as Krebs-Helmich cycle. The persons discovered this. Now the point you may have to remember here, very clearly. Even though ammonia is very toxic, remember one point. Very important point, because as concerned to this chapter especially, this ammonia will do some beneficial effects also to us. What is the one beneficial effects to us? Because this ammonia will help for acid-base balance in kidney tubules. So even though they are very toxic to our body, the one benefit they are doing to us, only I written here, they will help for this acid-base balance in the kidney tubules, or else we call it as, it will help for the buffer system. So remember this point also, very important. So it's very clear. So our excretory products is urea. That's why we call them as ureotelic. I written the examples: the humans, marine fishes, and the terrestrial amphibians. So these are the major points you may have to remember about this. Now moving on to the next nitrogenous based product we have to discuss. So ammonotelic is over, ureotelic is over. Actually, the next one is uricotelic. So listen everyone. Here what you have to understand, similar to the humans, this uricotelic organisms also, ammonia is the first major nitrogenous based product. But after that, in the case of humans, it will be converted to urea, but instead, in the case of the reptiles and the apes, that will be converted to uric acid. That's very simple. So listen, so everybody please understand. So your excretory product is uric acid. So what happened? What are the two examples? The examples are the apes or the birds, reptiles. These are the two examples. So here what you have to remember, in these organisms, ammonia is converted to uric acid. That's all. Instead of become the ammonia to urea, in this organism it become uric acid. But you might be observe that how the birds and the reptiles, the lizards, in the house lizards, how they excrete their products. You might be watched all the time. So you can see that how that will be the excreta, what is the shape of that. So you can see that some will be have the cream shape, or sorry, the creamy form. Some will be have the pellet form. Right. So there can be the different forms of this. Okay. So the cream, the pellet, these are the some kind of forms of this excreta. You might be watched in the case of the birds, especially the lizards. Also, it is just like a pellet. So that's very clear. So this types of this excretory system is very clear: uricotelic, ureotelic, ammonotelic. The one point we may have to discuss here, that is also very important. I would like to rub here. So what's the point you may have to remember here? Everybody listen. If you compare this ammonotelic, ureotelic, uricotelic, some comparisons we have to do here. The first comparison is about the toxicity. And the second comparison is about the water requirement. So listen, we can look into that. The first is talk about the ammonotelic. It is highly toxic. Highly toxic. The ureotelic is less toxic than the ammonotelic. While we coming into the uricotelic, it is least toxic. Water requirement, ammonotelic, maximum water. Minimum, here least. So that comparison also, I think you got it. The toxicity and the water requirement. Ammonotelic, maximum toxicity. The ammonia is maximum toxic. Urea is less toxic than that. Uricotelic even lesser than that. It's a minimum toxicity. So I hope this much is very clear. So no confusion in anyone. Now we can move on to the next part. Right, in the next part, what we are going to discuss it is, if you take before we start with our kidney, we have to see what are the different excretory structures in some animals. That's the part we are going to see. And what are the different functions they will do. The first thing we are looking into that, you might be heard about in the Animal Kingdom chapter, we have talked about it. the flame cells. The flame cells, otherwise we call it as protonephridia. They will mainly do the osmoregulation and excretion function. So listen here, everybody please look into this. The flame cells and or the protonephridia, they will mainly do the osmoregulation and excretory function. And you might be already know that which are the examples or which are the organisms you can see this particular flame cells. It's very clear, we have learned this in the animal kingdom chapter. That is, one is Platyhelminthes. The number one example. Now, what is the some annelids? Not all annelids, some annelids. Then rotiferous. Rotiferous are the some kind of the rodents only seen in the sea or in the water. Then the one more, it is Cephalopoda. So these are the organisms you can see in the case of flame cells. The flame cell as the excretory structure. I hope it's very clear. So the flame cells or the protonephridia, the main organisms you can observe, the Platyhelminthes, some annelids, rotiferous, Cephalopoda. Their functions are osmoregulation and the excretion. That point is very clear. Now look on to the next one. The nephridia. It's very simple. Nephridia in the case of annelids. They also will help for the osmoregulation and excretion. Very simple. Now, moving on to the next, the nephridia after that. After that, please listen. So the next one, it is the antennal glands or green glands. It is seen in the case of crustaceans. It is seen in the case of crustaceans. So then, it's a very common another one more, it is Malpighian tubules. Malpighian tubules. It is in the case of arthropods. We have learned one example for that, the cockroach example, we have learned it already, right? So please remember that. So, what we discussed so far, the excretory structures in the other organisms, what we discussed about it. One, we call it as flame cells, nephridia, then antennal glands or the green glands, then the last one is Malpighian tubules. So I have written all the functions also and the examples for that particular organisms. So these are the major part we may have to discuss about the other animals, their excretion. Now the later session of this class, we only discuss about the human excretory system, nothing more than that. In this chapter, that only we have to discuss. Right, everyone? It's clear. So I'm going to rub everything. Now listen. Now what we are going to discuss, the excretory system of the humans. So what are the main structures of human excretory system? Main structures. It's very important. One is kidney. Very, very important kidney. Then ureter. Then urinary bladder. And finally the urethra. So here the question, one question they can ask it. It's a very simple question. Among these excretory structures, which are the paired one, which are the unpaired one? It's a very simple question. You can easily answer. So this is the urethra. So these kidney and the ureter, they are the paired structure, no doubt. But these are only one. They are unpaired. They are not paired. Not paired structure. Yeah. So we have two kidneys. We have two ureters. That's a point you may have to remember. Yeah. That's very clear. Now we can look into the structures and its position. Right. The first of all, look into the position is very important. So always remember the kidney's position we are always telling with respect to the vertebrae because it is the easy approach or the easy approach into the kidneys. Only always we will talk with respect to the vertebrae. So with respect to the vertebrae, if you are looking, you can see that. Everybody listen here. We have how many vertebrae or which are the types of vertebrae? We have the cervical vertebrae, around C7, 7 vertebrae. Then thoracic region, we have 12 vertebrae. The lumbar region, we have the 5 vertebrae. Then sacral, then finally the pockets. Now here, what do you have to understand? Thoracic vertebrae is 12. So you can take it as T12. So this is T1. Here it is C1. Here it is C7. Now here it is, listen, here it is lumbar 1, lumbar 2, lumbar 3. So the kidney is exactly located in between the last thoracic vertebrae and the third lumbar vertebrae, like this. Right? So this is the position of the kidney. Right? So listen, everybody, this is the L3 position. So this is the T12 position. So the kidney's location in the both sides, it is in between T12 and the L3. This point, please remember. That's very important point. Now the location is very clear. Now we can look on to the next one. Very important next part. Now, what are the histology or what is the histology of the kidney? We have to look into. So before we start the histology, what we have to mainly understand, the kidney is having two songs. If you open the kidney, I'm just drawing the one kidney here. If you open the kidney, you can see that the two songs in the kidney. So the outer zone we call it as cortex, inner zone we call it as medulla. That point is clear, no doubt in that. So the outer cortex and the inner medulla. That's very clear, no doubt. And The junction between the cortex and the medulla, we call it as cortico-medullary junction. Right? It's called cortico-medullary junction. It's very clear, no doubt. Now, listen to the next point. Here's the point you have to understand next. This junction, we call it as cortico-medullary junction. The outer is called cortex, inner is medulla. So that's clear, no doubt. Now, the next point you have to really understand. If you look on to the outside part, it is slightly convex. You can see that it is slightly convex. But please understand, everybody listen to here. If you come inside of the kidney, the other side, the anterior side, you can check it. They are slightly concave. Right. This middle of this concavity, we call a name. So this area is convex. The inner part, this region is concave. So the middle of that concavity, we call it as hilum. A lot of importance to this hilum. What is the importance of the hilum? The center of this concavity, we call it as hilum. So what's the importance of the hilum? Everybody please understand that. Through the hilum only, the renal vein, through the hilum only, renal vein, renal artery, and nerves will endorse into the kidney. Here. So through this hilum only, the renal vein, okay, renal vein, renal artery, and this nerve supplies will endorse into the body. Sorry, in the kidney. Here, that one is really important. So please understand that. So renal vein, renal artery, and the nerves, they will pass through this hilum. So no doubt in that. Absolutely clear. Now, listen everyone, the next one. If you further understand this songs, you will realize that. So this is again the kidney. Right. So this is again the corticomedullary junctions. Now in this corticomedullary junctions, if you are looking, you can see small pyramid structures like this. Right. What is this pyramid structure starting from the corticomedullary junction? So this pyramid structure starting from the corticomedullary junction, we call it as renal pyramids. Renal pyramids, you can see that it will open to the another one sector, and it is called as minor calyx. Minor calyx will be open to a major sector. It's called major calyx. Major calyx will be open to this region, and it's called renal pelvis. Renal pelvis will open to the ureter. So make it very clear. So let me make it very clear. So this is the this dotted lines. It is the corticomedullary junction. So from the corticomedullary junction, there is a pyramidal structure. It's called renal pyramid. Renal pyramid will be opened to a larger space. It's called callus, minor callus. The minor callus will be open to little more larger region, and that is called major callus. Major callus will be open to the renal pelvis. Renal pelvis open to the ureter. So I would like to write that again this. So everybody please look into that. So there it is, renal pyramid. Right? Renal pyramid will be opens to the minor callus, minor to major, major to renal pelvis, renal pelvis to ureter. Right? That's it regarding the structure. Clear to everyone? No confusion on that. Now, you understand that. Now, what we have to learn it, we have to clearly understand one more point there. Now, if you look on to this kidney, you check this cortical and the medullary regions. The cortex to this medullary regions, they will be opened like a small columns. That columns from the cortical region to the medullary region, we call it as columns of Bertini. What is that structure called? Columns of Bertini. So what is the columns of Bertini? From the cortex to the medullary region, it will be opened like a columns. So from the cortex to the medullary region, it will be the kidney will be opened like a columns, and that columns we call it as columns of Bertini. That's a point you may have to remember. Now, please listen to the next point. If you keep this, this kidney in our body, in the two sides we have the kidney, the left hand side and the right hand side. And if you open that kidneys in the both sides, you will understand one thing. Kidney contains one of the major functional unit, and that functional unit we call it as nephrons. That only we are going to discuss in detail in the next part. So I think this histology part is very clear, no doubt in this. The renal pyramid, the minor callus, major callus, renal pelvis, ureter, and the columns of Bertini. Now, if you are very clear about it, we are going to talk about the functional unit of the kidney. So, what I said, what is the functional unit of the kidney? It's nephrons. Functional unit of kidney is nephrons. Do not forget that. So how many nephrons we have it in one kidney? Around 1.5 million. So please remember that point also. So the functional unit of the kidney is nephrons, no doubt in that. Now what we are going to do next to this, we will keep this nephrons inside the kidney. So after that, how they will be look like, we have to understand. All right. So if you keep this, the functional unit, that nephron, if you're keeping inside the kidney, how they will be look like, what are its different parts, that we are going to discuss. So everybody listen here. And just enlarge the kidney's diagram. Right. So what are the two zones in the kidney? One is cortex. Another one is medulla. That's clear. Everybody clear. Now, Listen everyone, if you keep the nephrons, so nephron, let me talk about it first. After that, I will come to this diagram because otherwise you may get confused. So now listen everyone, I'm talking about the nephrons first. What are the different parts of the nephron? We can just look into that. This is right. This is the one nephron. Now here, what do you have to understand very clearly? I would like to bring your attention. This we call it as afferent arterioles, and this we call it as efferent arterioles. For imagination. Now, after that, this afferent arterioles, they will be further break down into capillaries, and that we call it as glomerulus. This glomerulus will be secured. They will be protected by a cup-like structure, and it's called Bowman's capsule. Right. So, very proximal to the Bowman's capsule, there is another structure. It's called proximal convoluted tubule. After that, we have the loop of Henle. Sorry, this part. This we call it as this from the where the proximal convoluted tubule ends. From there to the up to the distal convoluted tubule, we call it as loop of Henle. Right. The loop of Henle, the beginning, we call it as descending loop of Henle. And the top one going up, it's called ascending loop of Henle. Now, after the loop of Henle region, ascending loop of Henle region, you can see one more convoluted part. It's most of the regions, and that we call it as DCT, distal convoluted tubule. Now, moving on to the last, and that part we call it as collecting duct. Right. So let me write about it. Everybody listen. What are the parts of the nephrons? So let me write that. So first one, afferent arterioles. So afferent arterioles, they will further break down, and that will form glomerulus. Okay. After the glomerulus, you can see that they will be again goes to the efferent arterioles. Right, that's very clear. Now, after that, you can see that PCT is there, descending loop of Henle is there, ascending loop of Henle is there, distal convoluted tubule is there, and finally the collecting duct. Clear, everyone? That's very clear. Now, here the point you have to understand, one point really you have to understand, that is regarding the collecting duct. For example, you consider this collecting duct. This collecting duct can be utilized by many nephrons. So let me tell about it very clearly. Regarding the collecting duct, one collecting duct is not the part of the individual nephron. That collecting duct can be the part of many nephrons. So let me talk about it. You take the one collecting duct, into this collecting duct, many descending distal convoluted tubules can drain, or it can be reach up. So that's the reason. Always remember, sometime we will tell it as a nephron does not have an individual collecting duct. So one collecting duct may be distributed for many nephrons. It's not the one nephron does not have the ownership of one collecting duct. It will be the many nephrons collecting duct. Remember that. That's a very important point. So that's very clear. Now, you understood the collecting duct. You understood about the nephron. What are the structures I have written here? Now, what you have to understand here, if I place this nephrons inside this kidney, how they will be look like, we have to understand. So first of all, as usual, we can just draw the afferent arterioles and the efferent arterioles, right? So here are the Bowman's capsule, everything. I will redraw it. Okay. Right. So this is PCT. Right. So the distal convoluted tubule. Right. This is the loop of hanging. Right. So this is our loop. Distal convoluted tubule. This is our collecting duct started. Right. So here proximal convoluted tubule, proximal only, right? Now listen everyone, while you're looking onto the diagram itself, you can understand many things. I would like to draw up here. So look onto this diagram and just place the nephrons in the kidney. Everybody can understand that. I just place the nephrons inside the kidney in the two different zones. So do not forget, this is cortex, this is medulla. So look onto the cortex region. What are the different parts located inside the cortex region? One is afferent arterioles plus efferent arterioles, then glomerulus, Bowman's capsule, then after that, this part DCT, this part PCT, then the upper part of collecting duct. Right? So the cortex region, if you are looking, what are things? Afferent arterioles, you can see that. Efferent arterioles, you can see that. Afferent, efferent, glomerulus, Bowman's capsule, DCT, then PCT, then upper part of the collecting duct. It's very clear. Now, while you look on to the medullary region, what are structures you can observe? It's very simple. Which are that? One is loop of Henle. It's very simple. LOH, loop of Henle. Second, it is lower part of collecting duct. Clear? Everybody understood? So, Which are the regions present inside the medullary loop of Henle? This region. This is loop of Henle only. Then lower part of the collecting duct. That also will be come under the medullary. So the placement is very clear. If you understand the placement, understand about the nephronic structure, its placement is clear, and you also understood the zones that will cover it. Now, what you have to listen first, next to this. So all of you make it very clear. So everybody make it very clear. If you take the glomerulus and the Bowman's capsule, that together we call it as Malpighian corpuscles. Point is clear where you are. Glomerulus and the Bowman's capsule. It includes the afferent and the efferent. Plus Bowman's capsule that we together we call it as Malpighian corpuscles. Or else you can also call it as renal corpuscles. So that part also please remember. So these are the main things you may have to remember about the structural placement or the nephrons. I hope it's very clear, these many parts, no doubt with anyone. Right. So now, what we are going to discuss next to this, listen everyone. Even though this particular nephron we have drawn here, it's a general structure, but all the nephrons are very similar. We have to, next session, the first one, what we have to learn is, what are the two types of nephrons? And also we have to understand the blood supply to this kidney. That's the next part we are going to see. So everybody please look into that. So listen everyone. The next one we are going to discuss the types of nephrons. That's the important one. The types of nephrons. Right. In the types of nephron, the first one we are going to discuss. So again, we require the diagram. The kidney. Right. So we have the cortical region and the medullary region. Right. And we require one more diagram here. Right. So here also the similar diagram, but structures will be slightly different. Right. So all of you please look into this. Here are also the cortex and the medulla. So this is the cortico-medullary junction. So if you take some nephrons, listen, their afferent arteriole formation that will be very, very close to the this is the afferent arteriole, this is the efferent. can take this. If you analyze this, and I will draw the diagram, the next one also, after that you will clearly understand. Right. So listen. So in the both diagrams, of the kidney, I placed the nephrons. But can you see any difference in that? So can you see any difference? Definitely there is a difference. We just look into this corticomedullary junction with respect to this corticomedullary junction. In the first diagram, you can see that they are the afferent arterioles are very close. Afferent arterioles very close to the corticomedullary junction. And in this, you can see that this is also corticomedullary junction, but you can see that here afferent arterioles, if you take it away from the corticomedullary junction, away from C and J. Am I right? So, what makes the difference in that? That's really important. If you look onto this kind of nephrons, their PCT, if you are checking, they will be short. You can understand from the diagram itself. But you take the loop of Hammond, it will be longer. And you take this diagram, their PCT will be longer, and loop of Hammond will be shorter. Right? Am I right? So where the PCT is long, PCT is shorter and the loop of Hammond is longer, that kind of nephrons, we call a special name. We call it as medullary nephrons. Medullary nephrons. The other one, the PCT length is more. PCT more length here. That kind of nephrons we call it as cortical nephrons. Remember one thing. Listen that point very clearly. I would like to bring that. In our body, 80% it will be cortical nephrons. Only 20% medullary nephrons. So you can see that medullary nephrons, it goes deep into the medullary region, and the cortical nephrons, you cannot see that they are not going very deep into the medullary region. Am I right? Everybody clear that? So you can very clearly observe here the cortical nephrons, they are not going very deep, but here they are going a little more deeper. That's clear. No doubt, no here. Absolutely clear. So no confusions. Now, the next thing what you have to understand about the how this afferent arterioles and the efferent arterioles, how that works. You have to clearly understand. So for that, what you have to learn is you have to learn the blood supply to the kidney. Many concepts we have to discuss based on this. It's very, very important. Now listen everyone. The question here, we have to understand first of all how the blood will be reaching to the kidney, how it will be reach up to the afferent arterioles. That we have to understand. Because then only the filtration process will happen. Am I right? Because first of all, the blood should comes to this afferent arterioles. So I would like to bring your attention into that. After that, I will discuss. So everybody listen to here. Right, everybody listen. The blood is coming into the afferent arteriole some way. We don't know, we are going to see that. The afferent arterioles will be further break down into the glomerulus. Then your efferent arterioles is that I will explain to you. But the first question we have to answer here it is how the blood will be reaching into this afferent artery. That's a afferent arterioles. That's the first part we have to understand. Here. So we will reach into that afferent arterioles and that we will start from here. So everybody listen. So everybody listen. I'm going to explain how the blood will be reaching to the afferent arterioles. So everybody please look into that. If you look on to the kidney is getting the blood from the, majorly they are getting the blood from the renal artery. But renal artery from there is getting the blood. So the what I am going to tell next it is, it may not be required for the NEET point of view, but you have to clearly understand that concept, how it come up. So listen everyone. So you know that we have the heart. So from this is connected with the aorta, right? Now, these aorta will be further divided and it will form abdominal aorta. So here itself we are starting. Now listen, abdominal aorta so listen everyone, first one is abdominal aorta. Abdominal aorta will supply the blood into the renal artery. Renal artery will supply this blood to the next artery, we call it as segmental artery. From the segmental artery, the blood will goes to the interlobar artery. From the interlobar artery, the blood will goes to the this arcuate artery. From the arcuate artery, the blood will goes to the interlobular artery. From the interlobular artery, the blood will goes to the afferent arterioles. This is the mechanism. Clear? So, it's starting from the abdominal aorta, then goes to the renal artery into the segmental artery into the interlobar artery, then arcuate artery, interlobular artery, and finally the afferent arterioles. So this is the way the blood will reach into the kidneys. No doubt in that. You don't have to worry about this particular arteries names and all. It's not required for your understanding. I just given. Now the next point it is you have to really understand. Once through the afferent arterioles, blood reaches to this division of the afferent arteriole that is called glomerulus. Glomerulus are the capillaries only. How the glomerulus we got it? By the division of, what is a glomerulus? Everybody please understand that. Glomerulus are the division of afferent arterioles. Clear? Now, after that, the blood will pass through this glomerulus. Many of the materials, please understand, many of the materials, they will further pass into the Bowman's capsule, then into the PCT, then into the loop of Henle and other regions. But some materials, they cannot pass through the glomerulus or the Bowman's capsule. What will happen to them? That particles, they will be goes to this efferent arterioles. Clear? Make it very clear. Through this arterial system, blood will reaches to the afferent arterioles. Once they reaches into the afferent arterioles already divided and form the glomerulus. The blood will pass through the glomerulus. Am I right? So the blood will pass through the glomerulus. By the time, what will happen? By the time, many of the materials can pass through the glomerulus, but some materials cannot pass through the glomerulus. They will pass into where? They will pass into the efferent arterioles. That concept should be very clear. So materials that cannot pass through the glomerulus, they will pass into the efferent arterioles. That point is very important. Which all material cannot pass it, that I will discuss later. But from the efferent arterioles, where it will go? From the efferent arterioles, the blood will goes to the capillaries. Right. This capillaries, as they are outside the tubules, outside the tubules means outside this nephronal tubules, we call them as peritubular capillaries. What we call for that? We call it as peritubular capillaries. So what is the peritubular capillaries? That concept should be very clear. Whatever the material cannot pass through the glomerulus coming from the afferent arterioles will reach into the efferent arterioles. Efferent arterioles will be further divided and will form peritubular capillaries. Peri means outside, tubular means nephronal tubules, capillaries means capillaries. So peritubular capillaries means outside the tubule region. What we call for that? Peritubular capillaries. Now listen everyone. Now from this peritubular capillaries, now listen everyone. From this peritubular capillaries, blood will goes to where? The blood will goes to, you can easily say that, it will goes to segmental vein. Segmental vein, where the blood will go? It will goes to the, sorry. So first, where it will go? It is starting from the interlobular vein. Interlobular vein, it is going into the arcuate vein. Arcuate vein goes to the interlobar vein. Interlobar into the segmental vein. Segmental vein again goes to the renal vein. Then inferior vena cava. Then it will goes to the heart. Right. So through the efferent arteriole, whatever the materials we are getting, that will goes again into this particular heart. That's very simple. So whatever the material is not filtering through this glomerulus, they will reach into the efferent arterioles. then through this renal vein and inferior vena cava, finally back into the heart. Clear? Everybody understood? So this mechanism should be very clear. This concept should be very clear. So actually, what are the materials that we cannot filtrate? It is actually we are not losing. We are again gaining back into the body through this peritubular capillaries or through this efferent arterioles. Clear, everyone? Understood this point also? No doubt. Absolutely clear. So the arterial system and the venous system of the kidney is very, very clear. Now, if you are very clear about this, what do you have to understand next? The next we have to understand about a structure called Vasa Recta that I'm going to discuss. Now listen everyone, I just need this diagram only. So what we are going to discuss here, the discussion about the Vasa Recta. So listen everyone. You take the justa medullary nephrons, you take the loop of Henle region, and in this, if you're taking this loop of Henle region, their peritubular capillaries, you take it. This peritubular capillaries will be like a U shape. Am I right? They will be like this. Right. So in the justamedullary nephrons, peritubular capillaries will surround the loop of Henle and it will be have the characteristic U shape. And that structure we call it as Vasa Recta. That's simple. Here. Everybody please understand. You take the peritubular capillaries surrounding the loop of Henle, peritubular capillaries surrounding the what? Loop of Henle, in which nephrons? Justamedullary nephrons. It will be have the characteristic U shape and that we call it as Vasa Recta. Remember one point here very clearly. Vasa Recta will be generally absent in the case of our cortical nephrons. It is very, very rarely occurred. So Vasa Recta structure mainly will be observed in the case of justamedullary nephrons. I hope it's very clear. The peritubular capillaries surrounding the loop of Henle, having the U-shaped structure, and it's called Vasa Recta. Point is clear, everyone? It's a very important structure in the case of justamedullary nephrons. It is very, very, very rarely seen in the case of cortical nephrons. Clear, everyone? Understood? Right. Now, if you are very clear with this, we can move to the next. So I think this part is very clear, no doubt in that. Right. Now, everybody listen. So listen, everyone. The next thing, what we are going to discuss, it is the urine formation. Okay, that only we are going to discuss. Right. So what we have to understand here, it's very important concept. Everybody listen. if you want to understand about the urine formation, first of all, you have to clearly understand about the blood supply to the kidney. That's why we discuss through the different arteries, it will be reach into the this particular uh, afferent arterioles we have discussed, and through the venous system it will back to the heart. That also we discuss. Now, what we have to understand, urine formation per minute. Then we have to understand urine formation per day. And what are the mechanisms coming under this, that we have to understand very clearly. We can look into that. So everybody listen that. First of all, what I am saying here is, we have to understand from the heart how much ml. So basically it should be red. So exactly how much amount of the blood will come from the heart into the kidneys? We have to first understand that. Listen, you know that 1/5 of the cardiac output will reach into the kidney. 1/5 of the cardiac output will reach into the kidney. So you know that we have learned already in the body fluids, cardiac output is equal to stroke volume into heart rate. So the cardiac output generally is equal to 5 liters or 5000 ml. No doubt in that, absolutely clear. So it's 1/5 means 1000 ml. Clear? No doubt in that, absolutely clear. So 1000 ml or the 1 liter of the blood is coming into the kidney in the every minute. But what you have to understand here, but this is connecting with the afferent arterioles. Now this is efferent, right? This is Bowman's capsule. This is PCT. All right. Now this is now listen everyone. The blood reaching into the afferent arterioles through the various mechanisms we already discussed. You just imagine 1000 ml if it is coming into the kidney, how much if you have to be filtered, we have to clearly understand. It's a very important point. If you take just remember this concept we have learned in the body fluids. You take the 100 ml of the blood. In the 100 ml, we have learned very clearly that in that 40% will be blood cells, 60% will be plasma. This concept we have already. Now you check in this 1000 ml, 40% it is gone. That is 40% means it's 400 ml, it's gone. Rest 600 ml only. That is plasma. But the 600 ml plasma is, just imagine, the 400 ml it is passing through the it will reach into the efferent arterioles. Then it will go back into the heart. We don't have to discuss that. Am I right? The 400 ml gone. So 400 ml, it is blood cells. That is the RBC, WBC, platelets. That everything will be gone into the efferent arterioles. It cannot be filtered. So that's why 40% is gone. The rest is 600 ml. In the 600 ml also, please remember, the plasma proteins and many other components present in the plasma, they also cannot filter. So after that mechanism, please understand, after that, only 20% will be get filtered. That means around 120 ml. So let me make it very clear. So what we learned, if you are getting the 1/5 of the cardiac output into the kidney, that is the 1000 ml. In that 1000 ml, finally, in each minute, we are getting only 120 ml of the filtrate only. That particular amount of filtrate only, it will pass into the glomerulus, then into the Bowman's capsule, finally reach here. It is just 120 ml. That is the 600's 20%. So where the 400 ml gone? 400 ml gone. It has the blood cells like RBC, WBC, and the platelets. The rest 600 ml, only 20% will be come as filtrate, because other things will be gone as plasma proteins and other components, because it contains the calcium and many other things are there. They will be reabsorbed back. So again, it will be decreased. So only 120 ml we will get. Now listen, for the better understanding and better calculation, easy calculation, we can take it as 100 ml. So I'm talking about the 1 minute completely to the together with the both kidneys. Together with the both kidneys, we will get 100 ml filtrate in every minute. When the fluid passing through the glomerulus and the Bowman's capsule, that time that fluid we call it as filtrate. So we will get 100 ml of the filtrate in every minute. Now listen up very clearly. Around 80%, it will be reabsorbed in the PCT itself. Together with the distal convoluted and the Bowman's, this loop of handling, around 15% will go. In the collecting duct, around 4% will go. That means, listen here, 80 plus 15 plus 4. That means 99% it will be get reabsorbed. The filtrate, that is 100 ml, passing through the PCT, it will reabsorb 80%. While they passing through the loop of Hamlin and the DCT, it will reabsorb 15%. Then when they passing through the collecting duct, it will reabsorb 4%. So total 99% it will be reabsorbed. That means out of the 100 ml, 99% reabsorb means the rest 1 ml only. That 1 ml only, the urine formed per minute. Right? We will get only 1 ml of the urine per minute. It can be 1.2 if you are taking the, or 1.00. You will get if you are taking it as 120 ml, exact 600's 20%. Then also you calculate 1 ml per minute. So now you just think, in 1 hour 60 ml. So in 24 hours or in 1 day is equal to 24 into 6. It is 1440 ml. You can take it as 1.4 liters, or you can take it as 1.5 liters as a round value. So 1.5 liters of the urine only you will form every day. But remember, how much ml of this glomerulus, how much ml they will filtrate? Actually, glomerulus will filtrate around 180 liters per day, right? 180 liters per day they will filtrate. But your final urine amount only, I just said that it is 1.5 ml. Clear? So 1.5 liters of the urine, if you are getting per day, the amount of urine you will get in every minute. In 1 minute, your urine output is just 1 ml. It is per day calculation. Per day, you will get 1.5 liters. Clear? Everybody understood? So in 1 minute, 1 ml, 1 day, you will get 1.5 liters. So what exactly showing this? The efficacy of our kidney, the reabsorbing capacity. Just imagine 180 liters per day they are forming. Filtration is happening through the glomerulus. So based on this 180 liters, if you are taking, it is 125 ml per minute. Exactly if you are looking. So then also please understand around 178.5 liters they are getting reabsorbed. Just 1.5 liters only excreting as urine. That is it's very sophisticated, even though they are evolutionary. It is so much sophisticated. So please understand this 178.5 liters of this filtrate will be reabsorbed. Just 1.5 liters will be excreted as urine per day. Now, what are the main concept you have to remember here? So we understood the benchmark is 1.5 liters. Even it goes to the 2 liters, 2.5 liters, we will say it as quite normal according to the patients. But someone crosses the 3.5 liters per day, the urine, if he crosses or she crosses 3.5 liters per day, then we call that as polyuria. Someone's their urine output is very less, that means less than 400 ml, it's called oliguria. And someone's the amount is less than 50 ml, we call it as anuria, right? Then someone's blood contain the blood, then we call it as hematuria, right? Hematuria, because most of the time hematuria time the RBC will be escaped through the urine. That's why that characteristic red color, the hematuria. So remember that someone got the pain during the urination, we call it as dysuria. So polyuria, oliguria, anuria, hematuria, and dysuria, right? That point is also please remember. So polyuria is the characteristic symptom of in two diseases. One is diabetic insipidus, and another one is in the diabetic mellitus. Both cases it can be happen, right? So please remember that point very clearly. So you understood the concept of this from the blood, how the urine will be formed up. But I did not say the mechanism how that will happen. That I'm going to say, right? So I just only said one thing. Urine formation, how that will be. The amount of urine formation only just spoke. So now what I'm going to discuss it is how that exactly will happen, right? So we have look into the next part. So we can look into the next. So everybody listen. The next thing we are going to discuss. So listen. So we know that there is a afferent arterioles, they will be keep on divides, they will form the glomerulus from that efferent arterioles, right? Then Bowman's capsule, right? Then PCT, then loop of Henle, and DCT, yes. Now listen everyone, I told you that the blood will coming into the afferent arterioles, then it will pass into the glomerulus, right? That's very clear. But some material, they cannot pass through this glomerulus. They will filtered back into the efferent arterioles, then into the veins, and will reach back into the heart. That everybody understood. Now what we are going to listen here, it is from the afferent arteriole, whatever the blood is coming, how they will be filtered through the glomerulus. What kind of mechanism it is there. And also we will understand why some of the materials cannot pass through the glomerulus. Why they are returning back to the efferent arterioles. That's the point we are going to discuss. If you're taking this glomerulus part, in the glomerulus and the Bowman's capsule part, if you are taking, there you can observe a filtration unit. What we have it in the glomerulus plus this Bowman's capsule part, there is a filtration unit. How this filtration unit formed up, we can see that. Okay. The first layer of the filtration unit, it is called endothelium of glomerulus. I hope it's very clear what is the endothelium. Everybody know that every capillaries, arteries, veins, everything, they will be covered by. So please look into this. You take a blood vessel, they will be covered by the squamous epithelium, and that we call it as endothelium. I think everybody know that. This is called endothelium. So similar to that, this glomerulus is also having the endothelium. That is the first layer from the outside. Then after that, there is another membrane, another layer. This is called basement membrane. So everybody listen. What is the second layer? We call it as basement membrane. What is this first layer? First layer is endothelium of glomerulus. Clear? Endothelium of the glomerulus. Now one more layer is there actually. So that layer is not exactly like this. They have slightly, they have some other shade. Right, they will be look like this. They are epithelial cells only, but their shapes are slightly different. Right. This one, this red color, I marked it. It's a epithelial cells only, the squamous epithelial cells only. We call it as podocytes. So actually, the podocytes are the squamous epithelial cells inside the Bowman's capsule. That only this podocytes. Podo means foot. So podocyte means foot-like structure. That's why we call it as podocytes. So the filtration unit, please understand the first unit it is endothelium of the glomerulus. Second one it is basement membrane. And the third one is epithelium of the Bowman's capsule. It's called podocytes. Now we can see that the blood will first pass through the endothelium of this glomerulus. Second, it will pass through the basement membrane. Third, it will pass through this podocytes. After that, it will reach into the PCT. Clear? Clear? Everybody clear this? So blood, it is coming from here, this side. It is passing through this. Next, it will pass through the basement membrane, then into the Bowman's capsule, then into the PCT. This is the PCT, just imagine. So like this, right? So that means the blood coming into the afferent arteriole and it will pass into the glomerulus, then into the Bowman's capsule, then into the tubules of the nephrons. But how many layers they are passing and doing the filtration? They are passing into the 3 layers. The number 1 layer from the outside, it is endothelium of the glomerulus. Second layer, it is basement, and the innermost layer is porocytes, the epithelium of the Bowman's capsule. That means that filtration is very, very efficient. So you can call it as it is not simple filtration. You just imagine you are filtering a coffee or something through the 3 sieves or the 4 sieves. It's not the simple filtration. Every particles will be just saved inside of that sieves. So this kind of filtration, we call it as not the just a filtration, we call it as it's an ultra filtration. Why we call it as ultra filtration? Because filtration is happening through the 3 membranes. That's why we call it as filtration, ultra filtration. So whatever the fluid we are getting after the ultra filtration, we call it as filtrate. So whatever the fluid we are getting after the ultra filtration, we call it as filtrate. I think that is also clear. Now, you take this endotheliums. This endotheliums, in between the endotheliums, you can see the many pores. So this kind of, as the endothelium having the pores, we call that endothelium as fenestrated endothelium. What we call that endothelium? Fenestrated endothelium. Clear? So I think these points are very clear. So ultrafiltration is very clear. What is the filtrate is clear, and what is the fenestrated endothelium is clear. Now the point you have to remember very clearly. Concept should be very clear. You can see that big gaps here. It's a fenestrated types. So it's having the opening, or that pores are very, very big. But then also why the many particles cannot pass? I told you in the previous part, the plasma proteins cannot pass. Blood cells cannot pass. Why? The answer is very simple. The very big cells cannot pass through this filtration unit. So let me make a diagram here. So these are the endothelium. Okay, these are the basement membrane, and these are the podocytes. It's very clear. Podocytes, basement membrane, and the endothelium. Even the big cells cannot pass through that. But some of the small cells also cannot pass through that, the small particles also. Because, listen everybody very clearly this. This is very important part. Actually, when the particle passing through this glomerulus and the Bowman's capsule, they have to cross 2 kinds of barriers. One is the size barrier. That means larger particles cannot pass through that. If some very large particle come, they cannot pass. But some particle, even they are smaller also, they cannot pass. The reason behind it is, apart from this size barrier, there is another one more barrier is called charge barrier. Charge barrier in the sense, listen, if you take this endothelial part, they contain so many negatively charged glycoproteins. So what will happen? What is the importance of this? When the negatively charged particles try to escape through this, they will not allow, because negative and negative, they will, if the same charge means they will repel each other. So they cannot pass it. So negatively charged particles cannot pass through this because in the endothelium of the glomerulus, they contain the negatively charged glycoproteins. So it's impossible to pass through that. So the endothelium of the glomerulus, please remember, they will provide the size barrier, at the same time they will provide the charge barrier too. That's very important point, never should forget. Clear, everyone? Clear, right? Now, if you are very clear this, I would like to give one more point here. The final 1 ml of the urine we are getting in 1 minute is the byproduct of, one is filtration, it's very clear. But whatever the filtration happened, we cannot get it as the urine, but many things will get reabsorbed. Am I right? But at the same time, many materials will be secreted into the tubules. Am I right? Many things will be secreted into the tubules in the different part of the nephrons. So you can say that the urine is the byproduct of filtration, reabsorption, and the secretion. Am I right? If you want to get the 1 ml of the urine, that particular fluid has to be, or the filtrate has to be gone through the filtration, the reabsorption, and the secretion. So you can say like this, as the filtration and the secretion, if you filtration plus secretion minus reabsorption is equal to the urine. Am I right? This is very clear. Whatever is filtrating, whatever is secreting, minus whatever is reabsorbing, the rest amount is called, or its difference we call it as urine. Very simple. So urine is by the mechanisms how the urines are formed up: the filtration, reabsorption, and the secretion. So filtration plus secretion minus reabsorption, you will get the urine in every minute. So in every minute, these mechanisms are happening, no doubt. Absolutely clear, everybody? No doubt, no? Right, so the urine formation is also clear, very clear. So we started with the urine formation, how that will come from the heart. Then 1 minute, how much ml will be formed up. Then we discuss about the mechanism behind it. That's a part we discuss. Right. Now we can move to the next part. So let me talk to the next. Now, what do you realize that now? So everybody listen here. In every 1 minute, the filtration is happening through the glomerulus. Am I right? Everybody make it very clear. Every 1 minute, the filtration is happening through this glomerulus every 1 minute. So how much amount of this filtrate will be come up? It's around 125 ml per minute. And per day, if you are considering, it is 180 liters per day. So we can tell it as the mechanism of fluid filtrating through glomerulus is called glomerular filtration rate, GFR. The amount of the fluid that will filter through the glomerulus in every minute. By the minute, you can calculate the day also. That particular mechanism or that particular filtration we call it as glomerular filtration rate. The rate at which the fluid will filtrate through the glomerulus, we call it as glomerular filtration rate. So it is 125 ml per minute and 180 liters per day. So remember, when the filtration happened, the filtration, that unit also you should have to remember. Now, we have a very sophisticated filtration mechanism. We have the sophisticated reabsorption mechanism and the secretory mechanism too. Now, we have to understand this mechanism. We have to prove that it is so much sophisticated. So how can we prove that? For that, we can take an example. For example, in your body, you just imagine there is a decreased GFR. That condition, our tubules of the nephron and the glomerulus, how they will react, we have to understand. So everybody listen. So before we understand that point, we have to understand one point very clearly here. After that, I will go to that. What is that one point? If you take the juxtamedullary nephrons, this is the cortical medullary junction. So in that perspective, I can tell you this is the juxtamedullary nephrons. This is the juxtamedullary nephrons. No doubt in that. Absolutely clear. But some juxtamedullary nephrons are not like this. Remember that. Okay, their structures are slightly different. Right. So listen, some of the juxtamedullary nephrons, this is juxtamedullary nephrons. Some of the juxtamedullary nephrons, if you are taking, this is afferent arterioles, sorry, this is efferent arterioles, this is afferent arterioles, and this is DCT. In some juxtamedullary nephrons, if you consider, there, listen, there, afferent arterioles and distal convoluted tubule, they will be joined together. Then that structure we call it as juxtaglomerular apparatus. What is that called? Juxtaglomerular apparatus. So in the case of juxtamedullary nephrons, their afferent arterioles and the DCT will be joined together, and that will form juxtaglomerular apparatus. Okay. This juxtaglomerular apparatus, if you are looking, they contain a specialized cells. We call it as JG cells, juxtaglomerular cells. This juxtaglomerular cells, they will produce an enzyme. It's called renin. Right. This much clear to everyone. So let me talk about it very clearly. We are talking about the juxtamedullary nephrons. In some juxtamedullary nephrons, their afferent arterioles and the distal convoluted tubules, they will be joined together and will form the juxtaglomerular apparatus, or the JGA. They contain a cells called JG cells, okay, or JGA cells, you can say. They will produce a enzyme called renin. Now, listen everyone, that's the point I'm going to discuss. When the GFR decreases, immediately who will come to know that? The JG cells will recognize that, or they will get the message. This JG cells will produce the renin. Renin, what they will do? Renin next mechanism is very brilliantly they will act. What is their brilliant act? Another one enzyme actually coming from the liver, it's called angiotensinogen. They will be released into this blood circulation. It's all the time it's there. So what will happen? This renin will convert this angiotensinogen from the liver. So I will just write it here. So what the renin will do? Renin will from where the angiotensinogen is coming? Angiotensinogen is coming from liver. This angiotensinogen, renin, what they will do? They will convert this angiotensinogen from the liver into angiotensin 1. Now, the next mechanism is angiotensin will be converted to angiotensin 2 with the help of an enzyme. It's called angiotensin convertizing enzyme. We call it as ACE. Actually, this angiotensin convertizing enzyme is formed in the lungs. All right, it formed in the lungs or pulmonary system. Now, this angiotensin, they will have the two way of actions. The one way, they will directly cause the vasoconstriction. Then, second way it is, they will stimulate the aldosterone. When the aldosterone stimulated up, what will happen? They will increase the reabsorption of sodium and water from DCT and collecting duct. When the sodium and the water reabsorb more, it is what will happen? That will goes to the peritubular capillaries. When that goes to the peritubular capillaries, again it will goes to the renal vein, then into the inferior vena cava, then into the heart. Then automatically what will happen? The amount of blood pumping from the heart, that is the cardiac output part, will increase. Then amount of blood will reach into the kidney also will increase. That means automatically the GFR will increase. Clear? So this is a mechanism happen if the renin angiotensinogen mechanism is get stimulated. So please understand this also we can call it as renin. We can also call it as ROS because renin angiotensin aldosterone mechanism, because aldosterone mainly from the adrenal cortex only. You might be remembering that. So you can also call it as RAS, renin angiotensinogen aldosterone mechanism. Also you can call it as RAS, renin angiotensin mechanism also. So by this mechanism, the GFR rate will increase. Clear, everyone? Understood this? So no confusion here also, absolutely clear. So how if the GFR decrease, how the renin angiotensin mechanism will increase, that we understood. So what is this juxtaglomerular apparatus also we understood. Now we can move on to the next part. So listen everyone, if we are very clear with this, we can move to the next. Now what we have to understand next is, listen everyone, The next it is the tubular system. We already discussed about it. So we can just look into the different parts. So here, this is afferent, this is efferent. Right. Now this is Bowman's capsule. Okay. Right. Right. Everybody listen. First of all, we can start with the proximal convoluted tubule. Proximal convoluted tubule, they have a convoluted part as well as a straight part. The straight part, we call it as pars recta. The convoluted part, we call it as pars convoluta. So listen, everyone. In the proximal convoluted part, listen, sodium, amino acids, glucose, bicarbonate, urea support, everything will get reabsorbed. They will get reabsorbed. Right. But remember, the H plus, K plus, and ammonia will get secreted in the case of proximal convoluted tubule. Now, moving on to the next part, here what you have to understand, one point you have to make it very clear. The glucose will be 100% reabsorbed, 100% reabsorbed in the PCT itself. So that's why you cannot see any kind of glucose in your urine, because it will be 100%, it will be reabsorbed. That's the reason behind it. Clear? Now, please understand that while we coming into the distal loop of Henle, you have to understand descending loop of Henle, you have to understand. In the descending loop of Henle, you can see that, everybody please understand, they are very permeable to the water. That means H2O will be get reabsorbed. So what is the character of descending loop of Henle? Descending loop of Henle, permeable to the water, but they are totally impermeable to the electrolytes. Totally impermeable to the electrolytes. Clear? But while we coming into the ascending loop of Henle, listen everyone, ascending loop of Henle, you can clearly understand they are permeable to the electrolytes. The ascending loop of Henle, you have to clearly understand they are permeable to electrolytes, but impermeable to the water. Water cannot move out. Right? That's very important point. Now, coming into the distal convoluted tubule. Distal convoluted tubule, it's a convoluted tubule only. So very few parts only, it's very straight. right? In the distal convoluted tubule, you have to clearly understand the selective reabsorption. That point is very important. Selective reabsorption of Na plus and the H2O. It's a selective reabsorption. So this reabsorption is very selective whenever it requires with the help of aldosterone. That's a part you may have to remember. Now, but in this case, in the case of DCT, you have to remember that many things will be secreted into the DCT. It's similar to the PCT, K plus, H plus, and the ammonia. Right. Now, coming into the last part, that is the collecting duct. Remember that one collecting duct for mini nephrons. Remember that point. Now, what all things will be get reabsorbed? The sodium, then water, no doubt in that. Then the one very special guest will get reabsorbed here. It is. It's nothing but the urea. All right. So they will be also reabsorbed here. But one point you may have to remember, here also secretion is happening. Secretion here, what will be happen? The main thing will be get secreted here. It is one is H plus ions, another one is K plus ions. These two things will be secreted. The Na plus H2O and the urea, they will get reabsorbed. Clear, everyone? So PCT, it's very clear what all things will be reabsorbed. Distal loop of handling, descending loop of handling, we learned that they are permeable to the water, totally impermeable to the electrolytes. Ascending loop of handling, permeable to the electrolytes, impermeable to the water. And the secretions, PCT, distal convoluted tubule, and this collecting that we have seen. Here it is H plus, K plus, and ammonia. Here also the similar, and here it is K plus and the H plus ions. I think it's very clear to everyone. No confusion here. Absolutely clear. Okay. So if you're very clear with this, we can move to the next part. Now the next one, everybody listen here. So kidney's main function, it is excretion. But also remember, they will help for the osmoregulation. So osmoregulation automatically it's a function of the kidney. So because while they're doing the excretion, automatically it's a part of this osmoregulatory system also. So now what we are going to see it as how the osmoregulatory system working in our body, that we have to understand. Because osmoregulatory function actually it's not monitored by kidney alone. We require the many help for that. So that part only we are going to see it. So osmoregulatory function in the body. So everybody please understand this. How the osmoregulatory function will happen in the body. Actually, first of all, you have to understand if any function want to be happen very smooth manner. First of all, body should have to detect that there is some problem. Then only we can smoothen the things. Or based on the problems, only body can act it. I'm very clear about it. So in the case of body, if you're looking, osmoregulatory functions will be monitored by three structures. Mainly the hypothalamus, second one is kidney, third one is heart. So actually how they will understand this hypothalamus, kidney and the heart, you have to very clear understanding about it. They can detect osmolarity changes. What is osmolarity? The amount of solute present in a solvent. That means if you take your blood as a solvent or the water as the solvent, how much solute are present, how much sodium, potassium, everything is present. Based on that, you can detect the osmolarity of your blood. So for example, to detect this osmolarity, you just imagine, for example, you just imagine it's very high. That will be detected by the hypothalamus, kidney and the heart. If the osmolarity is very, very low, that also can detect by this particular hypothalamus, kidney and the heart. That only we are going to discuss. So you understood what is osmolarity. For example, you just imagine, to understand this osmolarity, we have the Osmo Receptors. It is present in the different part of our body. So Osmo Receptors present in different parts of the body. Now, through this Osmo Receptor, the osmolarity can determine. While we understanding the osmolarity, what all things we will realize from that? We will understand the blood volume. We will understand the osmoreceptors will understand the ionic concentration, and also they can understand the blood pressure through this mechanism, through the osmoreceptors. So while the osmoreceptors detecting the osmolarity, they will realize about the blood volume in the body, ionic concentration in the body, blood pressure. Then this osmoreceptors, they will stimulate the hypothalamus. They will make the actions in the kidney and the heart. That's a mechanism really happening. So we can realize one thing like that. For example, you just imagine you are passing in the Sahara Desert. It's very hot. You cannot allow the urine too much gone out from your body because it will affect your body balance. So you will realize that the amount of solute in the urine will be very high because you cannot allow the water to go out. So osmolarity will be very high. So by the time, so please understand, by the time if your osmoreceptors detected, what will happen while you traveling in the Sahara Desert? Osmoreceptors will get stimulated. These osmoreceptors, they will stimulate the hypothalamus, hypothalamus will synthesize the one hormone corresponding to this, that is called antidiuretic hormone. So when this antidiuretic hormone released up, ADH, they will goes to the kidney. Kidney, which part they will act? They will act in the collecting duct, and they will reabsorb the water and the sodium. So automatically what will be happen? Your urine volume will get decreased, but your urine concentration increase. Why this body will do this mechanism while you traveling in the Sahara Desert? Because otherwise body will lose a lot of water in the form of urine. So that's why urine, they may make it concentrated, but at the same time urine volume will make it decrease. So by the mechanism of this ADH. But you just imagine another mechanism. After your thirst also, you are drinking too much water. Drinking too much water. So what will happen? That also will be detected by Osmo Receptors. Osmo Receptors detected. Then what will happen? They will stimulate the hypothalamus. But this time, hypothalamus will never stimulate to produce the ADH. They will inhibit the production of ADH. So when the ADH get inhibited, no reabsorption from the collecting duct. So the more water will be gone out. So what will happen? Increase urine volume, but less urine concentration. Clear? Increase urine volume, less urine concentration. Point, is it clear? So our body always will act based on this osmolarity. For that we have the osmo receptors. So how this osmo receptors will happen? They will work this. They will collaborate with the hypothalamus, kidney, and the heart. So in the previously we have learned one thing. Listen, when the GFR decreases, that will stimulate the renin angiotensinogen mechanism. In that also this is working. How? When the renin angiotensinogen mechanism, the GFR decrease, by that they will bring into the increased GFR. But always remember, body will check again this increased GFR. If it is too much, again another hormone will come from the atrium of the heart, and that hormone we call it as atrial natriuretic factor, or we call it as atrial natriuretic factor, ANF. They will cause the vasodilation and will decrease the GFR again. In the sense, they will maintain it. The increase, if the GFR, if it further increase, that will be decreased by this vasodilator, or the blood pressure increases, that will be decreased by this atrial natriuretic factor. So what we understood from this, whether it is increase or decrease GFR, or increase the decrease of the blood pressure, normally body will compensate it through many mechanisms. For that only we have seen this. So if the GFR through the renin angiotensin GFR increase, we cannot allow the further increase. We should have to limit it, balance it. For balancing, to decrease the GFR or to decrease the blood pressure, body will produce another hormone that is from the heart, the atrium of the heart. That's why we call it as atrial natriuretic factor. They will cause the vasodilation, thereby blood pressure and the GFR will come down and it will get balanced. That's very simple. So atrial natriuretic factor will cause the vasodilation. Clear, everybody? This much also very clear. Now the last part we are moving. So I hope it's very clear. So the very last part. So how the process, the urine will be gone out from our body, that we have to see it. the urine that will be moved out from or excrete out from our body, we call it as micturition. How this will be happen, we have to see it. So first of all, listen, everybody listen here. That is a kidney, right? So from the nephrons, every minute urine is coming out, then it will coming into the ureter, then into the, after that where it should go, this urine through the ureter, where it will come, it will come into the urinary bladder. Am I right? Then from there through the urethra it will go out. But you know one thing very clearly. So that point you know very clearly one thing. This is a urethra, this is urinary bladder. This is ureter, this is urinary bladder, and finally urethra. But in this mechanism, you know clearly one thing. When every 1 minute the urine are forming up, immediately the urinary bladder will not excrete out. They will first allow to come inside. It will store the urine for some time. Around 1.5 liters and all urinary bladder can easily store up. Now, what we are going to see it, whatever the urine formed from the nephrons through the ureter, it will reach into the urinary bladder. Now, once it reach into the urinary bladder, what will happen there? That only we are going to see it. Everybody listen to that. Right. So when the urine fill in the urinary bladder coming from the ureter, from the both ureter, what will happen? This urinary bladder contains the, listen everyone, this urinary bladder contains the Strux receptors. What they have it? Strux receptors. What is the use of this Strux receptors? When the urine filled up, this Strux receptors will get stretched up. When this urinary bladder stretched up, this Strux receptors will get stimulated. These Strux receptors are sensory in nature, so they will bring the information. They will bring the information to the brain. Okay, all these will reach into the brain. Right. Now, from the brain, this is sensory. So from the brain, what will come? The motor impulse will come. When the motor impulse come into the urinary bladder, what will happen? Urinary bladder will get contract. When the urinary bladder contract, please understand, in the urethral regions, two sphincters are present. One sphincter we call it as internal sphincter, internal urethral sphincter. Another one we call it as external urethral sphincter. So what they will do? When the contraction of the urinary bladder happened, so what happened first? Urine filled in the urinary bladder. Very clear. Stimulate the Strux receptors. So what will happen? Through that, sensory impulse will go to brain. Then from there, what will happen? Motor impulse will come. Then what will cause? Urinary bladder contraction. That's clear. So when the urinary bladder contracts, what is the next thing? Internal urethral sphincter and the external urethral sphincters, they will relax. Sphincters will relax. When the sphincter get relaxed, urine will come out, right? So no confusion, no here. It's absolutely clear. So first the urinary bladder will be filled up. So it will cause the stretch. It will pass the information to the brain. From the brain through the motor neuron, it will reach back into the urinary bladder. It will contract. So automatically the sphincters will get opened. Sphincters are circular smooth muscle we have discussed in the last class. So what will happen? Urine will move out. This urine generally will be very acidic, slightly acidic only, not very acidic, slightly acidic. 6.5 is the pH value, right? Because of the H plus ions secretion into the urine. That point also please remember, right? So that's very clear, everybody. Now we have discussed almost everything, and the last and final part. Listen, everyone. The last part. So listen, everyone. Actually, other than the kidney, some other organ structures in our body also can do the excretion function. So that excretory structures only we are going to see it. First one we are discussing here, it is if you take the lungs, what they will excrete? Definitely the carbon dioxide. Liver, what they will excrete? Toxins, excess vitamins, the cholesterol. So do not think that they are directly excreting out. They will help for the mechanisms. But the final excretion will be through the fecal matter and through the urine only. Toxins, excess vitamins, excess hormones, the bile, etc. Right. Looking on to the next one, that is the skin. So through the skin, a lot of water will be move out. Okay. Then sodium. Then sweat. Through the sweat, urea, lactic acid, sodium, etc. Then through the sebaceous gland, so mainly the hydrocarbons, the sterols, right? So these are the then sebum, definitely the wax, right? So these are the main products that can be excreted through the other body parts. Now moving on to the next. If at any point, if the kidney is not working, supposed to be at any circumstance, you got some kidney failure. By the time if the kidney is not working for the long time, you cannot survive. Because your toxic products like urea, the ammonia, every product sometimes will get accumulated. The body become very toxic. So under that circumstances, if you get any kind of kidney failures, your kidney is not working. What is a kidney failure? Your kidney is failed to work to remove the toxins. In that condition, sometime if you are giving the drugs or any other things, it may not be possible to bring back the patient into the normal status, or you cannot allow the patient to survive. So at that situation, what we will do it is, we have only the one life-saving technique, and that is called the dialysis. So generally, we will do the hemodialysis, right? That's a one life-saving techniques if the kidney failure comes up. Because in the kidney failure, what is the main problem? The urea amount will increase, other toxic products will accumulate. So in the beginning, we said that many toxic, many waste products, everything will get accumulated and become toxic. Okay, so the waste products cannot excreted out. That's a problem in the case of kidney failure. So the best technique you can make the survival of the patient, it's called the technique, we call it as dialysis. Why we call it as hemodialysis? Because it is happening through the blood. There is one more dialysis actually that is called peritoneal dialysis. That also please remember, there is one more type. It's happened with the help of peritoneal fluid. It's present in our this abdominal region. All right. Now, the material we are using for this hemodialysis dialysis unit, we use a material with a pores. That material we call it as cellophane. Remember that point also. So in this mechanism, blood will be taken through an artery and it will pass into this dialyzing unit. Then the blood will be filtered out as purified. Then it will again pass through a vein and bring back into this particular body. So let me make it very clear. So in the case of dialysis technique, so everybody please listen. So we will, the patients, that particular person, the blood will be taken through an artery. It will goes to the one dialyzing unit. In that dialyzing unit, some fluids will be present. So in that fluid, everything will be present similar to the plasma, except the nitrogenous waste. So what will happen? While the blood passing through this, all the materials, I told you already that the similar to the plasma, what all materials is that materials will be present in this fluid also, this dialyzing fluid. So by the diffusion mechanism, the nitrogenous waste products, that is the urea, uric acid, everything will be filtered out, or we will get, and rest every materials will get any excess sodium or potassium or the calcium, if it is there, that also will be filtered out. And at last, what will do? The purified blood, what we are getting, that will just pass through a vein and that will again reach back into the body of the person. Right. So this is what the simple mechanism of the dialysis, the hemodialysis. First, through an artery, the blood will goes to the dialyzing unit. Then inside of it, what material is present? Cellophane material is present. It contains the pores. While they're passing through the pores, every toxic material and the excess materials will be just filtered out, and we will get the purified blood. And that purified blood we will pass through a vein into the body. So while we passing the blood from the artery into the dialyzing unit, we will add the heparin to avoid the clotting. But while we passing the blood into the venous system, by the time we will add the anti-heparin. That point also please remember. Then the one very last part, the disorders of the kidney. Already we discussed one, kidney failure. The one more, it is glomerulonephritis. What is a glomerulonephritis? Inflammation of the glomerulus, we call it as glomerulonephritis. Okay. What is a glomerulonephritis? Inflammation of the glomerulus, we call it as glomerulonephritis. And the very last one, renal calculi. Renal calculi means kidney stone. This kidney stone mainly formed by calcium oxalate crystals. Sometimes uric acid crystals. In the case of gout, one disease yesterday we discussed in the locomotion, that disease also, this uric acid stones in the kidney can be formed out. So kidney stones, otherwise we call it as
renal calculi. And also please remember, when the urea increase in our blood, the condition we call it as uremia. And other nitrogenous waste also increase, then overall we call it as azotemia. So if the urea alone, we call it as uremia. If all the nitrogenous waste, we call it as azotemia. So that's all this chapter, the excretory system. So almost every point we have covered in this. So thank you very much. We'll meet in the next class with the next chapter. Thank you, everyone. Bye.